Hello everyone, my name is Brian. And today I'm talking about marine biofilms constitute a bank of hidden microbial diversity and functional potential. And first of all, we need to know what is biofilm. It's formed by bacteria, fungi, algae, protozoa, and metazoa. And its size is between about 100 and 200 micrometers. And it can produce EPS, and which plays important role in its structure and protection. And you can think EPS is the brick of the house. Okay. And how to form a biofilm? First of all, you need a organic surface coating, and you will attract some bacteria and stick on it. And more bacteria will be attracted and also stick on it, and it will produce EPS. And more some fungi, algae, protozoa, and metazoa also be attracted, and it will form a biofilm. Yes. And the pros and cons about the biofilms. We know about their cons. We know very about their cons. Yes, but pros we do not know. It's cons about the biofouling, biocorrosion, and bacterial disease. For example, is 那个 sorry periodontitis is a 牙周病. It's a bacterial disease. And we need to find what are their pros. So that's also one of our purpose. Okay, the purpose of this research, the first of all is revealing the diversity of marine biofilms and their functions. And the second is explore their environmental adaptability and their application of the biotechnology. Okay, we are going to data and methods. First of all, we are doing sampling. And the sampling, before the sampling, we need to cultivate it. And the cultivation site, we choose 8 sites. Sapelo Island, Red Sea, Hong Kong seawater, Yushih Bay is also in Hong Kong. And East China Sea and South China Sea. It has 3 sites in South China Sea, so the total site is about 8 sites. And the artificial surface for cultivation, we use a sterile PS Petri dish. And the PEEK, PTFE, PVC is the common artificial polymers. And then we used some metals, stainless steel, titanium, aluminum, and zinc. And this is for cultivation. And then we're going to collect them. We collect the biofilm we cultivated and the nearby natural biofilms, and we also collect the seawater samples and store them into the DNA storage buffer. This is for collection. And the second, we're going to extract their DNA and do metagenomic analysis. Before DNA extraction, we need to use lysozyme to break the bacteria's cell wall, break their entire structure to extract the DNA. And we use the TIA NAP genomic DNA kit. It's just a kit, yes, for DNA extraction. And then we are going to sequencing them. And the sequencing site is in the Novogene Bioinformatics Institute and Beijing Genomics Institute. They are all in Beijing, yes. And the metagenomes analysis shows that there are about 115 million reads per biofilm, and it's an average. And 130 million reads per seawater. And each read has about 150 base pairs. Base pair is the ATGC. Yes. is sequencing. And the third, we go into taxonomic profiling. And we need to download the OMRGC, their samples and their metagenomics. It's about 300 million reads. And what is OMRGC? It's a database for marine biofilms genes, and it's generated by a Terra Ocean project. You can think it's a dictionary for the metagenomes. And this is a comparison for our samples. And then we go into classification, profiling, yes. And we use the 16S MyTex, and also it's also the 16S rRNA gene sequences. And they are the reason, the 3 domains, 6 kingdoms, their classification is used the 16S MyTex. And we use the OTU to profiling them. What is OTU? It's a unit for recognizing and classifying microbes. OTU, for example, is, and we are all Homo sapiens, right? And in this classroom, we are just one OTU. But we don't use OTU because we already have a name, Homo sapiens. And there are too many kinds of bacteria. So if you wanna give it a name, it will spend a lot of time. So you use the OTU. And this is profiling. And we use the reads before the OMRGC and we sequencing. Then we need to give the functional genes a notation. And first of all, we use this 5 is the database for function classification. And we need to use this database for ARGS selection. What is ARGS? It's antibiotic resistant gene, 就是抗生素的基因。Yes. And then we going to do the BGCS identification and use this anti-smash. This is a software for doing this. What is BGCS? Is biosynthetic gene clusters, and they are used to biosynthesize bioactive compounds. And let's go to the results and discussion. You can, first of all, you can see the picture on my left-hand side. And you can see there are about 50, we only observe 50 OTUs in the seawater sample, and about 13,000 of OTUs in biofilm samples, and about 300 OTUs in the Tara seawater project. The right-hand side picture is important because you can, as I said, you can think that the red circle is a dictionary, but you see the blue part, it's out of dictionary. It means that there has many of the biofilm genes we are unknown. But why there has so many kinds of bacteria? We don't know why. First of all, the marine microbes exist in water rarely, and they proliferate rapidly and form a steady microbial community only in the appropriate environment. And the third, the traditional way to research microplanktons may not catch them. So there are so many kinds of bacteria we don't know, microbes we don't know. And the second result, it's about, first of all, we create a BCGC, which means a biofilm core genes catalog. And it's not quite important because we only want to show that these three databases is about the non-core genes. We already know, but they are about 61.1% genes we don't know. So there are so many that we don't know yet. Okay, about non-biofilm core genes which are related to biofilm adaptability includes that extracellular polysaccharide biosynthesis and DNA repair oxidative stress response and antiphobic resistance and osmotic and heat shock stress responses. And the meta-res database analysis shows that there are about 800 genes take part in 18 different kinds of antibiotic resistance mechanism. And the discussion is the microbes in biofilm may have the stronger adaptability and viability, which means the ability to survive. Microbes in biofilms have more antibiotic resistance than microplanktons, maybe because their EPS production. Okay, and this picture shows that the gene clusters and the different kinds of bacteria. And we only focus on these three gene clusters. Okay. PKS, NRPS, and PKS, NRPS. And what are they doing? PKS is used to synthesize the polypeptides, and NRPS is synthesize non-ribosomal peptide. And the third one is the hybrid of these two. And the paper mentioned that there are about 150 NRPS, 119 PKS, and about 20 PKS NRPS in this figure. And the similarity between the 85.6 of these three kinds of.
Can I ask about the 序凝作用, English? Flocculation. The formation of the biofilm and the change of the element between elements.
Does biofilm contribute to flocculation?
Okay.
Flocculation means the application of particles.
It didn't mention it, so I don't know what the influence is. Yes.
What does the Venn diagram in Fig 2 tell us about the overlap of OTU between biofilms and Tara seawater samples?
Oh, it's a non-species because they, as I said, you can think the red circle is a dictionary. So, and the blue circle is the current study, and the overlapping part is the we already know this species of the bacteria. Yes.
What is the 7748 mean?
It's the you can observe this also the OTUS, and you can observe you mean the gray part, right? Yes, it can observe not only in the new seawater samples. It can also observe in the biofilms and the Tara seawater. It's a Venn diagram.
First, I want to do a simple conclusion about the article, and I want to know what is the importance of the article. And at the 13 page, the graph, why you want to choose the three type of the in the statistic?
The conclusion is that it just say that it may have many application for medical development and genetic engineering. It's the simple conclusion. Yes. And what's the second question? We say importance, right? Yes, I think it's the same answer for the first question. Yes. And you say why choose these three gene clusters? It didn't mention it, but I think they will synthesize some of the new natural polymers, and maybe it's important for the medical development. Yes, and I'm not quite sure why it chose these 3 gene clusters.
Okay, thank you.
Okay, so from time to time, there were always some students who would like to challenge molecular biology paper, and I have to say it's a really tough task because most of us, we don't really understand molecular biology. Therefore, the speaker need to be first very patient. So he needs to explain what you are actually doing. And actually, I think the first part of the talk was good. And then the second is because this, you see, it's a Nature communication paper. So the content is too much for a 10, 12 minutes talk. So you need to choose which is the most important results that you want to present. So I think this one is really good, really nice. The second, you are talking about pros and cons, and later I saw that for the paper you didn't manage to present, you revisit the slide again. So I think there must be something you want to add because you left some empty space here. For the cons, I would suggest that for each of them, you can find some image to help us to visualize the cons of biofilms. That would be more interesting. And here, the purpose was clearly, very clearly stated. That was also nice. And I do appreciate that you spent quite some time explaining all these details. But I think this is nicely done, I have to say, this flow chart. So I now understand the paper better. But at the end, these kind of slides took you too much time because you didn't manage to your conclusion. So I think, for example, you were reading these, how many reads, etc. Perhaps these kinds of details can be taken out next time. You just tell us, because I think what's most informative for me is this. You did DNA extraction and then you do sequencing. That's all I need, you know? Okay, so these are all too detailed. I don't even know need to know they are in Beijing. That's not necessary. And then you only need to tell us that you go to some, you say, dictionary to understand the words you find from the DNA. That's really helpful. But still, these, I would suggest that you reduce the level of details, like the numbers, and then you just tell us in the general steps you are doing. So this is really to detail, but it's helpful. Then I also think if you have the notes here and you have space, you can make the notes larger. Go to your results. I hope that next time, let's go to that's the message you want to deliver at the end. You say you discover new antibiotics, enzymes, and rich genes. That's the key information, right? Therefore, if you stick to this key message, then you will know better what should you remove. For example, here, I don't think you need both of them. Okay. You can, because they almost say the same thing, nearly. Okay, you just take, I think this one is easier to understand. I would go for the easier one. And you tell, okay, there are so many new genes. Okay, so it's a world to be discovered. You can quickly go to, I don't know is it already regarding genes or not yet? Is it genes? Regarding their function already? Then you probably need to tell us can you read from here? Because you want to say there might be antibiotic resistance, where to look from this graph? Is it possible? You have to stick to your key message because it's a short talk. So you want to tell us there might be antibiotics, which seems to be the most valuable things. Please stick to this one. Because later you have these complicated, image. You have to think which one helps you to explain the key message here. And I would suggest that don't show all of the data figure. That's too much. You can take out at least one slide to make it into 12 minutes. Okay. And here he has this data discussion right after data format, which is also good. So this part is good. And I also hope that next time, because you have all these different figure results, here maybe you can tell us what is this about. You have space. Use your header. Maybe here is about diversity. Here is probably about functional genes and whatever. You use a short header to tell us what it is about. And next time when you were able to condense your methods and you have more time to explain your data, maybe you can take time to remind us what are these, because I already forgot. When you were here, I don't not remember what they are. You have to remind us. And then when you were talking about this, please use some simpler words. Use plain language. And then it's a general issue that when you show a figure, you don't need to show figure caption. You should use some guideline and use your own simple words to explain. And you don't need this. You don't need this. Okay? Okay, so I hope that next time you can make your whole talk into 12 minutes. Okay, good. Thank you.